Hello. So I've been working in longevity for the last 13 years, and I've been doing research on this topic, but most of you haven't heard about me because I was working in industry. So most of my research has remained secret, unfortunately. But each time I talk about what I do, and a lot of what I do has to do with finding new longevity molecules, people ask me, how do you find such molecules? And so my talk today will be about that. So I have other interests. I run companies now, but this is a very interesting point and I think very necessary since finding new longevity molecules is very much necessary. I don't have a click. There it is. Right. So when you think about finding new molecules, that's not probably what you have in mind. The classic way to find new molecules is by massive screening. This is the most common way. This is how the pharmaceutical industry is doing it. It's still done, of course, and it's actually the most, it's very efficient if you already have a very defined target. And this is also true for longevity, of course. But when you want to have an approach that is more based on finding new molecules that you don't have a specific target for, then you have to become a molecule hunter. So I don't have the right hat, obviously, but I've been, let's say, molecule hunter for a few years. And you have to be armed with a strong knowledge of evolution and ideally some good basis in chemistry as well. And then when you want to actually search for molecules, you have to find either the right environment to find such molecules, or to find the right organism, the right species. And I will talk about also the necessity of finding the right test, because screening is still necessary. So the importance of searching for the right environment to get new longevity molecules is, it's crucial in some specific cases. And the reason for that is that evolution has search for some environment will be beneficial for the apparition of some specific molecules. The easiest way for me to explain that to you is to tell you the story of how I found one of mine. So right after my PhD, I started in the lab of Miroslav Radman in Paris. And his interest was about extremophile organisms, bacteria, and how they manage to repair their DNA. That was his starting point. And he realized that actually the importance was not so much repairing DNA than it was protecting the enzymes that were actually repairing the DNA. So he switched his interest from DNA to protein and basically came to the idea that, okay, to be able to be resistant to stress in general, and of course now you see the relation with longevity, then it's actually very important to be able to protect proteins. So he asked me to search for molecule that would do that, protecting protein against stress in general. And back at the time, I was in Paris, and I was supposed to find extremophile organism in that environment. And that's not the most obvious one, because most of the extremophile that are described, you think about hyperthermophile, and that's different kind of environment. But when it comes to oxidation resistance, the possibility of, and that's what I was looking for, basically you can find that almost anywhere. So I dig into literature and I check what kind of natural isolate of extremophile bacteria were already found. And basically I found that some people were taking hot air balloons and collecting samples basically in the clouds and finding organisms there. And I didn't have a hot air balloon at the time, still don't. But basically, I came with the idea, okay, so all I have to do is take a plate for growing bacteria and coming outside on a rainy or a snowy day. It was February, so it was snowing. So I actually did that. I went outside. I went and picking up falling snow, well, not collecting it on the ground to avoid contamination with things that were already there. And then I irradiate this plate to make sure that I only keep the strong organism, getting rid of anything that is weak, because that was not what I was looking for. And from that plate, I found some interesting organism. So I, of course, went further in my research. I isolate one bacteria in particular. I grew it, I did extract of it, and started to test it for all sorts of ability that were interesting. Started, of course, with the most basic, like antioxidant potential, but not very exciting. Then I designed a test to be able to measure the ability of this extract to protect protein against oxidation, because that was my goal. And of course, then little by little searching for which molecule within this extract that had protective ability. So I found it, and I found a molecule by this way. And what was interesting is that this experiment, this part of Indiana Jones' work of going in the field, and I did it only once. I only went just once with that. That's very unusual for biologists to do an experiment just once, and it works. I mean, that's not even science, but okay. So yeah, and then we found that this molecule had amazing property. We were right with the starting hypothesis, and I will come back on that right after. This molecule, basically coming from an extremophile organism, have basically, how can I call that? Universal protective ability. So if I put it on a human cells and I put that cell under stress, the cell would be protected. And if I give it to a small animal model and I put it under stress, it will be protected as well. And if I also, of course, check for longevity, and also we show a really big increase of longevity on this organism. So the approach itself proved to be useful. And I remember a few years back in one of the Sense conference, they were talking about finding also actually similar approach of finding a molecule in nature through the right environment. They were searching for, I don't know if Aubrey is around, but he probably can confirm. They were searching for molecules to degrade cholesterol plate inside of the blood, and they went for cadavers. Their right environment were dead people in decomposition, because the idea was that within them you will find the bacteria that degrades the cholesterol. So it's actually very smart. Another approach that is possible, because there's no one solution fit all, obviously, is finding the right species. And I want to say finding the weird species. I'm very interested in nature and in animals, and the weirder they are, the more I love them. And okay, here I put a picture of some that you know about, not necessarily the one that I worked with, actually not at all. But I had another interest. So once I was done, and I worked for many years on this protein protection approach, I went to another topic. I had the chance to be pretty free in the type of research I was doing. Since I couldn't publish, basically I could do anything I wanted. And of course, there was a growing amount of evidence that being able to clean up cells by activating autophagy, proteasome, and other mechanisms like that was a very good way not only to slow down the aging process, but eventually to reverse it. So how do you find in nature molecules that activate this kind of mechanism? So at that moment, I was no longer in Paris. I moved to Split, Croatia, beautiful place by the sea. And I had a team also. Before I was alone. So with my team, we wander around. We started digging, well, we were by the sea, so we went in the sea. And we searched for animals that had described regenerative properties. And also for animal that has metamorphosis properties. So for example, larvae of insects, tadpoles that we went for the lake. So people in my team, they had fun, at least from time to time. Most of the time they were in the lab, to be honest. But once in a while we were going out, and that was like team building, but actually we were doing good work. And after collecting this, we did extraction. And so the logic behind it, I'm skipping some step here. The logic behind it is that these organisms have to go through massive changes. And usually these changes go together with activation of autophagy or localized or temporary hyperactivation of this cleaning mechanism. And so we did that. We collected these different samples. We did extraction. And then we tested it for activation of autophagy. We look at them. So we work on primary cells, primary human cell fibroblasts. And we measured the potential induction of autophagy. And again, we found it. So in both cases, I was pretty lucky, but I guess I have a right approach. But within less than, collection was maybe for just a few days. And then within a month, basically, we found at least the organism that was holding a molecule that was really activating very strongly autophagy. Then again, after the years, we saw that this extract that little by little was getting more and more pure down to the molecule was also able to have a rejuvenation effect on cells. So this was also confirming the initial hypothesis. There's a lot of research on that topic, so it was not really surprising. But at least we found one that nobody else had. So, right. So these are, like I said, the two main approach that it's not exhaustive, obviously, but the two main approach that I've been working with. And I need to talk about this part also because at the end of the line, to be able to find a new molecule, you have to have the right test. You have to have the right, you have to have something to screen on, even if it's not a massive screening, even if you are just screening for a few extracts of weird animals, you need to have the right test. And this is crucial. And basically that very often comes down to asking the right question. This is something that you probably heard about. And that's basically this talk is addressed to younger scientists, obviously. It's very crucial to ask the right question, and very, very often it comes down to using the right test. For example, for this protease protection approach, I had to come up with a test because there was none that would measure specifically the ability of a molecule to protect protein against oxidation. Something that was simple enough and could be done routinely, and that lasted less than 1 hour because I don't have patience anyway. So it had to be fast. And this came up with good results. When it came for autophagy, there are no, to my knowledge, tests that are so quick. You can hardly do that in 10 minutes. You have to grow cells, you have to. But so it was a different approach. But yeah, so this is one of the key elements. So my talk is voluntarily short. So if you have questions, and I know that lunch is coming, and I don't want to hold all of you, against the lunch that is coming. So I just want to finish with this question, and maybe this could be a start for a discussion about where will the next big longevity molecule come from. And I was really impressed by the talk on the bats. I like nature, like I said, and even so, not everybody thinks that this is the right approach. I think that this comparative biology approach still holds promises, and there's still a lot of things that we haven't found yet, and definitely a lot of environment, a lot of species that we haven't dig into, and so we're still at the beginning, basically, of the longevity molecules. We only have very few right now, and there's much more that are waiting for us. That's it. Thank you.